All right, hello. So this is the beginning of chapter 4, the tissue level of organization lecture. We're going to try doing this with a screen sharing here so that way you can see the lecture notes as I'm going through this. So we're ready to begin talking about tissues. Tissues are cells working together. Those cells that are going to form those tissues must have some sort of common embryonic origin, like the same cluster of cells is going to make that same type of tissue. Histology is collectively the study of a particular tissue type. This is what you do if you were sending off a tissue sample from a biopsy to have it looked at. Is it cancerous? Is it normal? Is there a malformation? That's what you're going to be doing, is sending that to a histologist that studies those particular tissue types. So there are 4 basic tissue types in a human: epithelial, connective, neural, and muscle tissue. Epithelial tissue covers surfaces like the interior of your blood vessels. Your skin is an epithelial tissue. It lines hollow organs, cavities, ducts, and forms glands when cells sink under the surface. So that could be either an endocrine gland like adrenal gland or pituitary gland and secrete something, or it could be an exocrine gland like a sweat gland or an earwax gland. Those are all types of specialized epithelial tissue cells. Connective tissue holds stuff together, as its name implies. Can also store energy. Fat is an example of this. Some of your immune cells are types of connective tissue. Muscle tissue just shortens. It's got one job, and that is to shorten in length, and that produces some sort of movement. Nerve tissue conducts signals that allows you to sense things and allows you to respond. So all nerve tissue does is conduct those signals. So what we said earlier is that if you have certain types of tissues, those have to come from certain embryonic tissues. The terms for those are germ layers. Like a germ is what something grows from. It is the endo, meso, or ectoderm, which is the inner, middle, or outer layer of, as you are developing from that one cell and you make a bunch of cells, and those bunch of cells start to differentiate, they make some layers. Depending on where those layers came from, whether it was endo, meso, or ecto, that will develop into different tissue types. So you get epithelial tissue from all 3 germ layers, connective tissue and muscle from the mesoderm in between, and nerve tissue from the ectoderm or the outside layer. So this is all really interesting how this forms, and that has direct impact on how your tissues are spread throughout your body. So one of the things we said when we were talking about a tissue is that if you were going to go to a histologist, you would have you may have a biopsy done. That is, there might be a mole, and that mole might not look normal to the dermatologist. They might remove that tissue and send it to a histologist. That is called a biopsy. That is what they would use to diagnose whether that mole was malignant or benign. Is it cancer? Is it not? You can't just slap a big chunk of a mole down onto a microscope and look at it. You have to slice it thin and then be able to look and see the actual cells that are making up the tissue. So that's just a common thing that you would do in a biopsy. So let's today begin talking specifically about epithelial tissues and some general features of all epithelial cells. So epithelial cells are packed very tightly. Now this makes sense because they're supposed to line and protect things. So if you're talking about skin, you want your cells to be tightly packed with one another, and they're held together very good. The cells set on top of an underlying layer that anchors them, called the basement membrane. You might see this on your coloring sheet. They have a free upper surface. They're exposed to an open area, like the inside of a blood vessel would be your apical surface, or the outside of your skin would be the apical surface. Your epithelial tissues are avascular. They do not have blood vessels running through them. Your skin, your actual epithelial tissue on the outer part of your skin, has no blood vessels in it. Now it has it below it, and then the nutrients must diffuse up into that skin. You have good nerve supply in your epithelial tissues, allows you to feel. and they divide really rather rapidly, which if you get an injury, it allows you to heal rapidly because you don't want to be left without some sort of protection. The basement membrane is what anchors that cell down to the underlying connective tissue. So if you look at this image that I've kind of hovering over here, here are our single layer of epithelial cells. Okay, they're open to wherever up here is your apical or your free surface, and then we anchor to the underlying connective tissue. This is made up of the basal lamina, or base layer is translated, and your reticular laminated, or the twisted layer. And so your epithelial cells will release these really strong collagen fibers from its basal lamina, or its base. And the underlying connective tissue will release this reticular lamina, or these reticular fibers, which are kind of bent and crooked. Now, this is really interesting. You've got these fibers from up here, and you've got these bent fibers down from below. Together, they hook together and form the basement membrane. Think of this like cellular Velcro. Those protein fibers interlock, and it links them together. So this is a really strong way of fibers interlocking. So it is like a protein Velcro to hold cells on top of other cells. That really kind of makes sense. So that's those big words kind of hide the idea, but they really just link together and anchor down that lining. Like, it would be kind of bad if your skin wasn't totally attached all over your body. It'd be loose or something. That'd be kind of weird. So it is good that you have this basement membrane to anchor your epithelial tissue onto whatever it is it's supposed to protect. Your different types of epithelium. There are different types based upon what they do. You either have covering or lining epithelium, like skin, or you have glandular epithelium, something that forms a gland and then produces some sort of secretion, like a sweat gland or adrenal gland or thyroid gland or all the other glands that secrete stuff in your body. So let's take a look first off at covering and lining epithelium. And let's look at how it is classified based upon how it is arranged. Okay, there's two different things to look at. You look at the shape of the cell and you look at how many layers. So the first part is arrangement of cells into layers. Does it have one layer or does it have more than one layer? So one layer is just referred to as being simple, cells one layer thick. This would be like the lining on your inside of your blood vessels. It's lined by one cell thick. Stratified is many cell layers thick. Your skin has many, many layers of cells all stacked up to make your skin as a whole. There is another type called pseudostratified. That prefix pseudo means fake or false. So it looks like it's stratified, but it's really not. It just means all the cells don't reach the outer surface. It looks multi-layered, but it's not, okay? So we look at one layer or multiple layers. Then we look at the shape of the cell. So first off, is it flat? Then we call that squamous. That would be like your cheek cells that we looked at in class. Cuboidal are cube shaped, that's a nice term, and columnar are tall columns. You also have another type called transitional that they can stretch. That would be like lining of your bladder because your bladder stretches as you fills with urine or empties of urine. And so those will actually change shape, which works out good for them. So let's go through and let's look at some different classifications. So first off is simple squamous. So single layer, simple, squamous, it's flat. So here you can see a single layer of flat cells, right? It's still anchored to underlying connective tissue with the basement membrane. This can line blood vessels. It can line body cavities. It's really thin, so things can still diffuse through it because you want oxygen and water and nutrients to diffuse through that if you're talking about blood vessels. And the cells are all in direct contact with each other. So here's an example. This is inside of your body cavity. These are the cells that line it. And this is in your intestines. And you can see this lining of flat squamous cells, single layer, lining your internal intestines. So you can absorb nutrients through them. Simple cuboidal is one single layer, and they're cube shaped. These are primarily located in these looped tubes that make up your kidneys that you diffuse, like you're diffusing water and other things out of your bloodstream in your kidneys, and then you collect it and then you urinate it out. And so that's one of the primary areas that you would find simple cuboidal cells. Here you can see that loop of Henle, which is the loop, and you can see these cells which are roughly cuboid. They're not perfect cubes, they're just roughly equidistant. And so you can see the nuclei stained red, and then this is the lumen, the open area. So this side would be the apical or free surface here and there, because this is open. And then your basement membrane would be that dark stained connective tissue underneath that. Non-ciliated simple columnar is you've got columns, you've got microvilli on the top. Remember that microvilli were projections of the cell that increased surface area.